kids. So Gregor Mendel, we always start with when we talk about heredity. It's called the father of genetics. He was a monk who lived in the 1860s and studied in an abbey in Austria. He was the first person to analyze patterns of inheritance and deduce the fundamental principles of genetics. He studied garden peas because they were easy to grow. They came in many readily distinguishable varieties, are easily manipulated, and can self-fertilize. There's a little TED Talk at the bottom about Mendel's pea plants, if you guys want to watch it. Heredity is the transmission of traits from one generation to the next. So these are all just kind of random vocabulary words we are going over at the beginning of these notes because we're going to be using these words throughout the notes quite frequently. Genetics is the scientific study of heredity. A character is a heritable feature that varies among individuals. A trait is a variant of a character. Each of the characters Mendel studied occurred in two distinct traits, an either-or situation. Traits with only two options are now called Mendelian traits, after Gregor Mendel. However, there are plenty of non-Mendelian traits that we will also talk about. So we know things like eye color and skin color, and even hair texture. Those aren't either-or traits. You can have a mixing of those. But we do have some traits that do occur either or. Freckles is one of them. Widow's peak is one of them. Earlobes, free earlobes or attached earlobes. Those are either or traits that you inherit from your parents. And those are called Mendelian traits. And the first half of the notes is about those traits. And then the second half is about the non-Mendelian traits. In the Abbey Garden. So an abbey is a place where monks and nuns and priests would go kind of live off by themselves. That's if you don't know what an abbey is. Mendel created purebred varieties of plants and crossed two of these different purebred varieties. Hybrids are the offspring of two different purebred varieties. And when we look at the kind of family tree throughout Mendel's experiments, the original generation is called the parent generation or the P generation. When you get to the first generation offspring, we label that F1, second generation is F2, and so on. So he crossed purple flowers, a pea plant with purple flowers, to a pea plant with white flowers as the original parent generation. And he found among the first generation, all plants had purple flowers. There wasn't a mix between the two. There wasn't any white. They were all purple. And then when he fertilized that second generation to each other, he got the next generation, the F2 generation, and he got a ratio of 3/4 of the plants have purple flowers and 1/4 of the plants have white flowers. And he did this over and over and over again. He was kind of OCD about it. So that 3-to-1 ratio was pretty consistent among these flowers. So 4 hypotheses came from Mendel's experiments. He dubbed these a monohybrid cross, or he didn't dub them a monohybrid cross, but scientists over the years did. Mono, we've talked about before, means one. So he was just looking at one trait while crossing these plants. And that first trait was the color of the flowers. So the alternative versions of genes are called alleles. So the gene is in your DNA, and then the allele is kind of what you see on the outside. Eye color, there's 4 different versions in that picture, but there's other versions that we know of. There's a darker brown, a lighter brown. Blue can be light or dark. Gray can have either hints of green or hints of blue in them. Either way, the alternative forms of all of that are called alleles. Second hypothesis, for each inherited character, an organism inherits 2 alleles, one from each parent. An organism is called homozygous for that gene if both alleles are identical, and we use the same letters when we write them down. Now, all of you have taken regular biology. Most of you have taken it with either myself or Miss Ibekwe. And in that class, we went over homozygous, heterozygous, dominant, recessive. We went over all of those. We know the prefix homo means same. So that is a genotype made of the same kind of alleles, either both big or both small. We call an organism heterozygous for the gene if the alleles are different. And again, we still use the same letters, but we use different versions of that letter. We use one big A and one small a. And we'll use all kinds of letters, but A is just what we start with for the example. You can see in the genes at the bottom, they're using B. And they're homozygous if they're the same version of the B, both capital or both lowercase. They're heterozygous if you have one of each, one capital and one lowercase. And these two words, homozygous and heterozygous, are going to be very important when you are learning about Punnett squares. Number 3, if 2 alleles of an inherited pair differ, then one determines the organism's appearance and is called the dominant allele. Again, we use capital letters for the dominant allele. Dominant, you can think of stronger or more powerful. So we use the big letters. The other has no noticeable effect on the organism's appearance, and it is called a recessive allele. We use lowercase letters. And then in the picture, we have the three different genotypes, the 3 different combinations of genes. And we use various versions of homozygous, heterozygous, dominant, recessive. So if you have 2 big letters, we use big letters for dominant, and if they're the same letters, it's homozygous. So we call that homozygous dominant. We have 1 of each. Hetero means different, or different versions of that letter. So we just say that is heterozygous. And then on the right, for the white flower, you have 2 small b's, 2 of the same small b's. Homo means same, so that's homozygous. And they're small b's, so that is recessive. Again, these are going to be very important concepts going forward when we're doing Punnett squares, when you do your lab this week, when you do your test this week. You have to know how to use those words, homozygous, heterozygous, dominant, recessive. And these were words that you were taught when you were freshmen once upon a time. So this should just kind of be a refreshing your memory about these words. Gametes, sperm and eggs, carry only 1 allele from each inherited character. We went over meiosis last week. Meiosis cuts the number of chromosomes in half. So each parent only has 1 to contribute to the next generation. Meiosis makes the gametes. The 2 alleles for a character segregate or separate from each other. During meiosis, they do it randomly. And this statement is called Mendel's law of segregation. So from all of Mendel's work, they named some laws after him. Punnett squares. This is a diagram that illustrates genetic crosses and allows us to determine the probability of certain offspring outcomes. 2 types of information can come from Punnett squares. You can figure out somebody's potential genotype, which is the organism's genetic makeup, and that's why we use the letters. We can also figure out the organism's potential phenotype, which is what it's actually going to look like, what their physical appearance is going to look like. So there's an example of a Punnett square there. We'll go through them in more detail, obviously. But when we talk about the genotype, we're talking about the letters. In this example, it's using Ys, big Ys and small Ys. And then the phenotype, the physical appearance, would just be the color. Those are pea pods for Mendel's pea plants. And are the pods going to be green or yellow? That would be the physical appearance that we're talking about. Homologous chromosomes. We already said homo means same, logous means structure. So homozygous is talking about the letters of a genotype. Homologous is talking about the chromosomes. So this is a pair of chromosomes with the same structure. And this is where the alleles are found. The different versions of the same gene at the same location on a certain chromosome. If you took Bio134 last semester with me, we talked about the location of certain genes on chromosomes a lot. Test cross is a purposeful mating between an individual of a dominant phenotype, but unknown genotype, and a homozygous recessive individual. So in Mendel's case, he would take purple flowers. Purple flowers, you know, they have at least one dominant gene, so one capital letter. But that second letter was up for grabs. So to figure out what that second letter is, he crossed it with a recessive plant, which is a plant with all white flowers. So in that first generation, if all of the offspring came out purple, you know that that original plant that had the unknown genotype had two dominant Ps, two dominant alleles. But if half of them came out white, then that purple plant was heterozygous with one big P and one small p, one dominant allele and one recessive allele put together. So this is just the same example with dogs. You have two dogs, a black one and a brown one. They look like Labradors. Brown is recessive. And they crossed it with a black dog, where they knew one of his Bs of his coat color, because it is a dominant, black is the dominant coat color. And they had to determine, was he homozygous dominant or she? Don't know which is which. Or was that black dog heterozygous with one big and one small? If he was homozygous dominant, then all of their children would have been black. But if he was heterozygous, then half of their children would be black, half of their children will be brown. Or their puppies, not their children. So Mendel's principles, when there are only two trait options, dominant and recessive, this is when we call them Mendelian traits, and they apply to the inheritance of many human traits. Like I said, freckles, a widow's peak. A widow's peak is kind of that point that a hair can come to in the middle. And then earlobes. Some people's earlobes are free and kind of hanging, and then some are a little more attached. And you guys will have all of these this week in both your labs. Wild type traits. So dominant traits are not necessarily normal or more common. When we're in the lab or when scientists are actually out in nature, when they describe these, they use the term wild type. So wild type traits are those seen most often in nature and not necessarily specified by dominant alleles. Blood type is a good example. Blood type O is the most common blood type, but it is a recessive allele. So blood type O would be considered a wild type trait. A family pedigree shows the history of a trait in a family and allows geneticists to analyze human traits. We went over a lot of pedigrees last semester in Bio 134. It's a series of circles and squares. Circles are always female, squares are always male. If they are filled in, it means they have whatever trait the pedigree is talking about. A lot of the times they do this with diseases. If they are half filled in, they're called carriers, and they're heterozygous for whatever trait they're talking about. In this pedigree, they're talking about sickle cell anemia. That is a recessive trait, and it's an either or. Either you have it or you don't. Many genetic disorders show simple inheritance patterns and are controlled by simple genes on autosomes. Most are recessive. There are some dominant disorders. Huntington's disease is a dominant disorder. Dwarfism is an actual dominant disorder. So individuals who have the recessive allele but appear normal are called carriers. That's when those circles and squares are half and half. They're heterozygous for the disease and they have one good allele to mask the diseased allele. So this is just an example. Hearing. This is how deafness is passed on. So sperm, the dad contributes one dominant allele and one recessive allele that goes on top of the Punnett square. Eggs, the mom contributes the same, one dominant allele, one recessive allele. And then they cross. So if you don't remember how to do a Punnett square, you get one from mom and one from dad. And then you get one from mom and one from dad. So you always mix the alleles in each square. One from mom, one from dad, and then one from mom, one from dad. And then you have to know how to interpret the Punnett square. So in this Punnett square, you have one child that's going to end up homozygous dominant. This child is homozygous dominant, and his hearing is going to be normal. These two children are heterozygous with one big D, one small d, and their hearing is going to be normal, but they're going to be called a carrier because they carry that disease gene and can potentially pass it on to their offspring. And then 1 out of 4 children between these two parents is going to end up deaf, who cannot hear at all. So you have to be able to put the Punnett squares together and then interpret what the children can potentially look like or what their characteristics may be. Some human genetic disorders are dominant, like I said. Achondroplasia is the official name for dwarfism, the official sciencey complicated name. The homozygous dominant genotype actually causes death of the embryo. Thus, only heterozygotes have the disorder. And the homozygous recessive children you'll see in a minute, what their phenotype is. Huntington's disease is another one, leads to the degeneration of the nervous system. And it doesn't usually even begin until middle age, until 40-ish. So people go their whole lives and then all of a sudden at 40 start developing these symptoms and they have no idea why. And then they have to go to the doctor, get their DNA tested, and then figure out, oh, you have Huntington's disease. So that would be a question. Do you want to know your DNA? I know we talked about it last semester, those home DNA kits. If you're young and you get it done, you can find out if you have the gene that's going to potentially develop into a disease later in life. Would you want to know that? Would you want to be prepared for that? So this is the achondroplasia or dwarfism example. So here, parents, you have one normal parent and one dwarf. These are the parents' genotypes. So dad is normal. He has 2 small d's. Remember, this is a dominant disease. So if he doesn't have the dominant allele, he does not have the disease. Mom has the heterozygous, and then their potential 4 children are in the middle. So they don't have 4 children at the same time, they have 1. So one child has a 50% chance of being heterozygous and having dwarfism. And then they have a 50% chance of being normal. So human genetic disorders that are controlled by simple recessive dominant patterns: albinism, cystic fibrosis, PKU, sickle cell, Tay-Sachs, and then dominant disorders: achondroplasia, Alzheimer's, Huntington's, hypercholesteremia, where you have excessively high cholesterol and it's passed down through generations. So today, many tests can detect the presence of disease-causing alleles. Most genetic tests are performed during pregnancy. Amniocentesis collects cells from amniotic fluid. Chorionic villus sampling removes the cells from placental tissue. And then genetic counseling helps patients understand the results and implications of genetic testing. So what kind of a chance would you give your child to have these certain traits, whether they're just regular traits or actual diseases? So skip that one for now. And then now we go on to exceptions for Mendel's laws. And this is just an example. So incomplete dominance is when traits blend and you get the mix between the 2. This happens sometimes. This happens with skin color is the best way to explain it. If a dark person and a light person have a baby, that baby is going to end up, their skin color is mixed. And then codominance is when both traits are shown individually. That'll make more sense when we talk about blood types. So incomplete dominance. Some patterns of genetic inheritance are not explained by Mendel's laws. So we call them non-Mendelian traits. In incomplete dominance, F1 hybrids have an appearance between the 2 phenotypes. So in your lab this week, you are going to have a question about a cross between red flowers and white flowers and how some of their offspring end up pink. And like I said, in a human example, skin color is the best thing to illustrate incomplete dominance. Blood typing. Blood typing is a few different ones. So blood typing is controlled by multiple alleles. Blood types in humans involve 3 alleles of a single gene. A and B are both dominant. And for some reason, we use I with a little superscript A and then I with a little superscript B. When letters are above like this, they're called superscripts. And then lowercase I is the recessive allele, but this equals blood type O. So type A is for people with two of these. It can be either IA, or the IA paired with a small I, because the dominant overpowers the recessive. Type B is the same, but with B's. Type AB is when a person has both. Mom gives them A, dad gives them B. They are called blood type AB. Both of them show. And then blood type O is the two recessive alleles. I don't know why we can't just use O, they use I's. But the letter refers to two carbohydrates designated A and B that may be found on the surface of blood cells. So multiple alleles, you have 3 alleles that control 4 different blood types. Matching blood types are critical for safe blood transfusions. If a donor's blood has a certain carbohydrate, A or B, that the recipient does not have, the recipient's immune system will produce antibodies as a mean to fight what it sees as foreign cells. This will cause the donor blood cells to clump and potentially kill the recipient. So your blood cells clump in the wrong artery or vein, it cuts off blood supply to certain areas. If it cuts off blood supply to the heart, that's when people have a heart attack. If it cuts off blood supply to the brain, that's when people have a stroke. So blood clots inside your body are not a good thing. So if you ever need a blood transfusion, it's really important to get the same type of blood or to get type O blood. Type O is the universal donor. It doesn't have any of those surface proteins to cause that clumping. So the 4 blood groups involved 3 alleles of a single gene, like I said. IA for the ability to make substance A, B to make substance B. I does not make either substance. Each person inherits 1 allele from each parent, making 6 possible combinations. If the person inherits both IA and B, both alleles are expressed equally, neither is dominant over the other. And this is called codominance. Co just means both. Both are dominant, both get shown in the genotype. Pleiotropy is when one gene influences several characters. Example is sickle cell disease. If a person has a gene for sickle cell disease, especially 2 genes, one from each parent, it results in abnormal hemoglobin proteins and causes disc-shaped red blood cells to deform into a sickle shape with jagged edges. These sickle-shaped cells get stuck in blood vessels. That's what causes the blood clots and potentially heart attacks and strokes if they get clotted in the wrong place. Polygenic inheritance, the additive effects of 2 or more genes on a single phenotype. Eye color, skin color, height. So not only is skin color incomplete, it's also controlled by more than 1 gene. Poly means many. Eye color is the typical example that we use and that we talk about. So it's controlled by more than 1 gene. So you need a bigger Punnett square. You guys are going to be working on these bigger 16 Punnett squares in your lab this week. But you can see a variety of different colors for eye color depending on the parents, ranging from dark brown to very, very light blue. And it all depends on the 4 different alleles and how they're interacting with each other. The role of environment. We went over this a lot in Bio134 as well. Many human characters result from a combination of both heredity and environment. Environment can include things like nutrition and exercise. Epigenetics is the study of the field of genetics, of cellular and physiological phenotypic trait variations that are caused by external or environmental factors that switch genes on and off and affect how cells read the genes instead of being caused by changes in the DNA sequence. If you need a review of epigenetics, Neil deGrasse Tyson is always my favorite. Does a great little summary of it. Linked genes, alleles that start out together on the same chromosome, would be expected to travel together during meiosis and fertilization. They're located close together on the same chromosome and tend to be inherited together. Remember, during meiosis, chromosomes can cross over and switch information. So the closer the genes are together, the more likely they are inherited together and not to be crossed over and traded to the other chromosome when that happens. And then crossing over can of course separate linked genes, but the closer they are together, the less likely they are to be separated. They produce gametes with recombinant genes. That's supposed to say genes, the second gametes. And they produce offspring with recombinant phenotypes. The percentage of recombinant offspring among the total is called the recombination frequency. So scientists can actually calculate which genes got switched over and how often by studying offspring. Not necessarily human offspring. Normally it's easier to see in smaller organisms that are quicker to multiply. So just an example, you have a pair of homologous chromosomes, A and B. That person's gametes or sex cells go through meiosis, crossover, so that the parental gametes, you have 2 original parental gametes in the end and then 2 recombinant gametes in the end. Sex determination. Humans have 23 pairs of chromosomes, 46 total. 22 pairs of autosomes that control 99% of our characteristics, our physical and personality traits, and 1 pair of sex chromosomes. In general, girls have 2 X chromosomes, boys have 1 X and 1 Y chromosome. Last semester, we went over different examples. People can have 3 X's, people can have XXY. People can have an X and a half. And there's just an example Punnett square to determine gender. And this works for most of the population. There are always exceptions. We went over a lot of those exceptions last semester. We did a whole discussion about it. About the women who had their metal stripped because they made too much testosterone or something else. Sex-linked genes. The sex chromosomes contain specific genes related to the many different characteristics, not just male, female. A gene located on one of the sex chromosomes is called a sex-linked gene. Most are found on the X chromosomes. The X chromosomes are bigger and just have a lot more information. Color blindness, hemophilia, which is a blood disorder, muscular dystrophy. Those are all disorders linked to the X chromosome. And those are recessive disorders. There's an X-linked dominant disorder called hypophosphatemia, which a person doesn't process phosphate the right way. The important thing that it's linked to the X chromosome and it's a dominant disease. So Punnett squares for X-linked genes. When determining sex-linked disorders, you have to keep the X's and Y's from the parents and then link the letters for the gene to the X. So again, this is where we call them a superscript. So whatever this B stands for, we'll say color blindness. I like to use B's for color blindness anyways. So you have mom up here. You put her gene for color blindness linked to the X. So she is heterozygous. For her, she has one big B, one small b. Dad only has one X, so he only has one B. The only reason the Y matters is to determine whether the offspring is a boy or a girl. So when you're doing Punnett squares for sex-linked disorders, for most of the time it's X, only the X's should have the letters linked to it. There's a question about an X-linked disorder on your lab this week. There will be a question about X-linked disorders on your test this week. And so the allele for color blindness is recessively linked to the X chromosome. So for this Punnett square, how many girls are there? What's the probability of having a girl? And then how many of those girls will be colorblind? What about boys? What's the probability of having a boy? And then what's the probability that boy will be colorblind? You kind of have to take this half and half because you have 50% chance of having a girl, 50% chance of having a boy. Then you have to figure out what their B's look like. What do the girls' B's look like? Are any of the girls going to be colorblind? And what about the boys? The boys only have the one X, so they only have the one B. They don't have anything to mask it if they have a recessive gene.